Good morning, students. Last class we have studied about the cell organelles, that is mitochondria and other membrane bound organelles. We have also studied about the plastids, in which I told you that this class I am going to discuss about the chloroplast in detail. The diagram of chloroplast is important for you all. It is very important. Again I am repeating that in class 11th you will not get diagrams to label, but you have to draw. So in point of view of drawing, labeling and drawing of chloroplast is very important. So let's start with the explanation of chloroplast. Majority of the chloroplast of green plants are found in the mesophyll cells of the leaves, and they vary in shape. They could be lens shape, they could be oval, they could be spherical, or they could be even ribbon like. Their number varies from 1 per cell to 20 to 40 per cell in the mesophyll. Like there are number varies, like there is 1 chloroplast in Chlamydomonas per cell, whereas if I take a green alga, you know the green algas are very long. They have they vary in size, so they can have 20 to 40 chloroplast per cell. Again, because it is a membrane bound organelle, and I have already discussed that they are not interconnected, so we will study it differently. So it is like mitochondria, it is also a double membrane structure. The space within the inner membrane is called the stroma. The space inside, suppose this is the membrane, the space inside it is called as stroma. And a number of flattened membranous sac-like structures are present inside it, which is called as thylakoids. The thylakoids are arranged in a pile. We have studied that also. They are arranged in piles, like we stack the coins one on top of the other, and this stack or the pile is called as grana. They are called as grana or granum as singular. There are flat membranous tubules called stroma lamellae connecting the thylakoids of different grana. If there are 2-3 piles, the channel which is connecting these piles is called as stroma lamellae. Right? Am I clear? Now, they enclose a lumen, an empty space. The membrane of the thylakoids, the inside of this membrane encloses an empty space which is called as lumen. This stroma, that means the inner part, the stroma, it contains many enzymes which are required for the process of photosynthesis. They even contain circular DNA molecules and ribosomes. Again, similar to mitochondria, they contain these DNA molecules or ribosomes because they are capable of self-duplication. They can reproduce, they can produce one more chloroplast. Chlorophyll pigments are present in the thylakoids, and the ribosomes could be 70S or 80S kind of ribosomes. Now, when I am talking about ribosomes earlier, when I am talking about endoplasmic reticulum, I told about ribosomes that they can be attached to the endoplasmic reticulum and can give them a rough look. So it's called as rough endoplasmic reticulum. But when studied alone, we found that it is a dense circular particle, and they are composed of RNA, protein, and these are not surrounded by any membrane. They have RNA, ribonucleic acid, and protein, complete proteins. The eukaryotic ribosomes are 80S, while the prokaryotic is your 70S. What do you mean by 80S and 70S? S stands as a subunit, or we can say a unit through which we measure these ribosomes. This is the sedimentation coefficient. That means they are indirectly the measure of density and size. Both these 70S and 80S ribosomes are made up of two subunits. The 70S is made up of 50 subunit plus 30 subunit, and the 80S is made up of 60 subunit plus 40 subunit. We are measuring the density of ribosomes with the help of S, which we call as Sevdur's unit, and or we can call it as sedimentation coefficient. Each is composed of 2 subunits. 70S is composed of 50S plus 30S, whereas 80S is composed of 60S and 40S. Now we come to another part which is called as cytoskeleton. This cytoskeleton is nothing but it is a filament-like structure, proteinous structure present in the cytoplasm, and together collectively they are known as cytoskeleton. They are involved in many functions like mechanical, for mobility, for maintenance. They could be called as, in some cases they are called as microfilament, microtubule or intermediate fiber. Among these we have cilia and flagella. It's like hair-like outgrowths. This is the membrane, and from here we have the hair-like outgrowth which we called as cilia or flagella. You remember we have flagella in euglena. It's also a structure like this coming out from the, and Chlamydomonas we have many cilia present. The slipper shoe shape, we have cilia on the surface. So they are the outgrowths of cell membrane. Cilia is a small structure, whereas flagella is a long structure. Both work for the movement of the cell or even carrying the microorganism from one part to another. The prokaryotic bacteria also possess flagella, but they are structurally different from that of eukaryotic flagella. When we study under the electron microscope, we found that cilia or flagella shows almost similar kind of structure. They are covered by a plasma membrane, and inside the plasma membrane, suppose if I take this as a cilia and this is the plasma membrane, inside this plasma membrane the core is called as axoneme. What is it called? The axoneme. The core is called the axoneme, and in this axoneme there are many microtubules, tube-like microtube-like structures running parallel in long axis. Small tubes running parallel in long axis. The axoneme usually has 9 pairs of doublets of radially arranged microtubules. 9 pairs in doubles, right? They are arranged in 9 pairs of doublets radially arranged of microtubule. Such an arrangement of microtubule is referred to as 9 plus 2. 9 bundles of 2 each, so 9 plus 2. The central tubule are connected by bridges and are enclosed in a central sheet. They are covered by a sheet-like structure. Now, 9 radial spokes and the peripheral doublets are also interconnected. So what we can say is that prokaryotes have 9 plus 0 type of made up of flagella protein, whereas eukaryotes have 9 plus 2 type of made up of tubulin protein, in which 9 are central and 2 are peripheral. Now we come to centrosome and centriole. What is centrosome? You have already studied that centrosome is an organelle which is a bag-like structure which contains 2 cylindrical dot-like structures, right, which we called as centrioles. So this bag is called as centrosome. Again, this is found in animal cell, not in plant cell, and we have the centrioles present. Both centrioles in a centrosome lie perpendicular 90 degree to each other. They don't lie parallel, they lie 90 degree to each other. And it's a cartwheel kind of a, means they are 90 degree to each other. These centrioles we have studied is responsible for providing spindle fibers during cell division in animal cells. One more thing, these centrioles also form the basal body, the base from which the cilia or the flagella arise. They are responsible for that also. So they have basically two functions. In unicellular or in lower organisms, they give rise to cilia or flagella, whereas in higher organisms they give rise to spindle fiber, which help in division of the chromosomes, which we called as spindle fibers, right? Now we are going to discuss about the nucleus, this which is called as director of the cell. That means it helps in all the functions. Nucleus, in one word, we call as director of the cell. It was discovered by Robert Brown. We have studied all these things, but we will study it in detail over here. The nucleus is basically stained by basic dyes. The nucleus was given the name chromatin because inside we have the chromatin fibers which are present. So this was the name was given by Fleming. If we see the interface nucleus, then we can see the proteins or the chromatin or the nuclear matrix, whatever you call it, is very dense. And if we see in electron microscopy, we see that the nuclear envelope, this nuclear membrane or nuclear envelope, is all consists of two membranes, and the space between these two membranes is called as perinuclear membrane. It forms barrier between the material present inside the nucleus and that present inside the cytoplasm. So the nucleus is a double membrane structure. The space between the two membranes is called as perinuclear space, and this nuclear membrane, it stops the prevents the entry and exist of material from inside the nucleus to outside into the cytoplasm. The outer membrane, if we talk about the outer membrane, I said there are two membranes. The outer membrane is in continuous with the endoplasmic reticulum, and sometimes it also bears ribosomes on it because they are responsible for protein synthesis. Some of the places you may find that the two membranes are fused together. We call these fused path as the nuclear pores, so that the materials can pass from here to there, passes through this. The movement of RNA and protein molecules takes place in both the direction from this nuclear pore, what we will call it as nuclear pore. And from this nuclear pore, movement of RNA and protein takes place in both direction. Normally there is one nucleus per cell, but there can be more than one nucleus. Some mature cells may even lack nucleus, like RBCs do not have nucleus. The RBCs of mammals do not have nucleus. Right? Now, the inner matrix, the inner part of this inside the nuclear membrane is called as nucleoplasm. What is it called? Nucleoplasm, which contains of nucleolus and chromatin. The content of the nucleolus is continuous with the rest of the nucleoplasm, and it is not a membrane bound structure. It is not a membrane bound structure. It's just a dark hole nucleolus. It is just a continuation parts, and here actually the RNA synthesis takes place. If the larger, the more numerous is the nuclei, the more actively it is continuing with the protein synthesis. When we have studied the different stages of cell divisions, we see structured chromosome in place of the nucleus. This chromatin contains of DNA and some basic proteins called as histone. Inside this we have the chromatin structure, which finally give rise to the chromosomes. A single human cell had approximately 2 meter long thread of DNA. That means there are 23 pairs of chromosome. Now let's talk about the chromosome. There's a primary constriction which we called as centromere, on the sides of which a disc-shaped structure which is called as kinetochore is present. Based on the centromere, we have the different structure. We have studied that: metacentric, submetacentric, acrocentric, telocentric. If it is present in the center, it is called as the equal arms, then it is called as your metacentric. If it is present on one side, then acrocentric. If it is present not in the between, then it is called as telocentric. Sometimes a few chromosomes have a secondary constriction somewhere, which is a non-staining secondary constriction, and this is called as satellite chromosomes. It's called as satellite chromosome, small fragment like, and we call it as a satellite chromosome. Right. So with this we come to the end of the chapter, which is cell. Hope you have understood. You have any problem, you can put on your queries by watching the video. I may give you few questions which has to be done in your notebook. Have a good day.